Welcome back to the Pharma Forum podcast. In this episode, I speak with Dr. Evan Floden, CEO and co-founder of Seqera, which aims to empower scientists with modern software engineering and data analysis tools built on an open science foundation. The conversation touches upon how the pandemic served as a catalyst for a revolution in genomic surveillance for tracking pathogens, and how the learnings from that period hold so much more potential if barriers are lowered for bioinformaticians. Tune in, let us know if you have any comments, and as ever, thank you for listening. Welcome to the Pharma Forum podcast. This is web editor Nicole Raleigh, and today I will be speaking with Dr. Evan Floden, CEO and co-founder of Seqera, a data orchestration and genomics analysis company. Genomic surveillance tracks viruses using genomic sequence data, a process to decipher the genetic material found in an organism or virus. The pandemic served as a catalyst for a revolution in genomic surveillance for tracking pathogens. The technology proved vital in aiding understanding of the evolution of and spread of virus in real time, to inform public health measures, ultimately accelerating drug and vaccine development. However, the full potential of genomic surveillance in public health labs remains locked away due to its high barrier of entry. Currently, complex workflows and requirements for specialized bioinformatics capabilities limit who can effectively utilize this technology, and there remains an urgent need to make it more accessible and adaptable for broader public health beyond pandemics. So it is today that Evan and I will be discussing how genomic surveillance has evolved since then and how the barrier of entry can be lowered to support future bioinformaticians. Seqera was founded in 2018 to empower scientists with modern software engineering and data analysis tools built on an open science foundation. Leveraging the deep knowledge of high throughput analysis and modern software engineering gained from building Nextflow, the Seqera Cloud and Seqera Enterprise platforms have also been created to make data-intensive research scalable, flexible, and, importantly, collaborative. Seqera has been used by major pharmaceutical and life sciences companies, including Genomics England, for optimizing their bioinformatics research processes so as to unlock valuable insights from genome data. To begin with, though, Evan, perhaps you could tell listeners a little bit more about yourself and your journey to doing what you do today with Seqera.
Okay, thanks a lot, Nicole. Thanks a lot for having me on the podcast. So a little background on myself. I was originally very interested in bio growing up, so through adolescence, studied biotechnology, joined a lab in my undergrad and had the opportunity to work on some yeast genetics. And at the time, we were looking at how we had different yeasts, with each had different genes removed from them, sort of looking at the interactions that could take place with sort of genomic interactions there, as well as how different chemicals were sort of affecting different genes there. And so I guess I've got a little bit of a bug for the computational side at that time. I then got the opportunity to join a startup which was developing medical device, which was made of the forestomach of sheep. And really through that experience, saw what was required to in some ways bring a product to market with FDA approval for that product. And the company eventually went public. At the same time, I saw the sort of trials and tribulations that are involved with spending so much time in the lab at the bench, and wanted to go much more on the computational side. So my background in sort of bioinformatics, more specifically, started when I moved to Europe. I did a master's degree in Italy, and then had the opportunity to spend some time in Cambridge in the UK at the Sanger Institute, working for RFAM. And some of the listeners may be aware of some of the resources there, like PFAM, where we essentially have a full collection of all of the proteins which exist from sequencing data, and then got the opportunity to do a PhD in Barcelona on multiple sequence alignment, which is really the underlying material that is used in many of the things for development of phylogenetic trees, as well as some of the more interesting sort of new stuff, which is all the rage at the moment with regards to things like AlphaFold and the three-dimensional structure prediction, which comes from all of this sequence information and then some of the other tools that have been applied from that. I guess I sort of studied in Barcelona. My PhD was originally supposed to be on multiple sequence alignment, and that's sort of how I sort of started. At the time, though, the kind of work was very computationally intensive. So we were using lots of different sort of big, large cluster machines and running this large computational analysis. And at the desk next to me, I sort of happened to have my now co-founder, Paulo, who was really responsible for helping us run this large-scale analysis. And his background was very different than mine. He comes from as computer scientists, and saw the opportunity there for how we could allow scientists to run these kind of large workflows using more modern software engineering tools that came about. And that's really the origin story of Nextflow. And this sort of predates Seqera by about 5 years or so. So this is 2013, 2014. And it was really around the time that these kind of workflow engines became really popular and sort of started off. So there's Nextflow. There's another very sort of popular one that some listeners are aware of called SnakeMake. And what these tools do is they allow scientists to take individual components. Maybe you've got scripts that you've got in the lab. And maybe you've got some tools that you're taking from GitHub or places like Bioconda, pulling all of these scientific tools together and then allowing you to run these things at scale, both in the cloud, but also in clusters and develop on your laptop, etc. I guess the key piece is that the sort of why that came around at that time is driven primarily from the large scale analysis that was just required. So as the advent of next generation sequencing is kind of really geared up, and the requirements around how much is required to analyze, combined with a few kind of interesting pieces on the tech side. So what we saw very early on was the ability to combine things like container technologies, so using Docker, which can encapsulate all the dependencies, the ability to use Git, which I think going on to the piece on genomic surveillance, it allows scientists to all work on the same code base in a collaborative way so that they can contribute code, and they can comment on it, they can work on it, pull requests, etc. And that's really was a large piece of the basis for what happened during the pandemic in terms of that kind of collaboration, where many of the world scientists sort of down tools and were able to kind of work on both in the lab, but also on the software side. And finally, the kind of more obvious one is around cloud and the use of cloud for this kind of large scale analysis, which is a trend which sort of continues. So Nextflow started in this manner. It was an open source project. It was placed on GitHub. We got sort of slow traction over time. Realized in about 2018 that there was really an opportunity where we had larger pharma companies and organizations reaching out for support, and then really took the opportunity to form Seqera as part of that. And from Seqera, then we've been able to build out what we call the Seqera platform, which is really a kind of full stack application, which goes beyond just the workflow execution, beyond just the pipelines, and more kind of on the data management piece as well. And that's really been leveraged quite significantly, both in pharma in terms of drug development and in biotech, but also in terms of diagnostics. So it's becoming more routine for this large scale analysis to be involved in diagnostics, as well as also in terms of public health, which is kind of the main topic that we're going to be discussing today.
Thank you, Evan. Gosh, that's been quite a journey, a very full journey at that. Thank you for walking us through it. So if we turn to our main theme, as you were alluding to towards the end of that there, on genomic surveillance and the use of GET in particular during the pandemic, how have things changed since the pandemic itself, Evan?
So what I sort of saw very early on was at least some of the work which was being done by some of the pipelines that we've been involved with. One of them in particular was a protocol which is called the Arctic Protocol. And prior to the pandemic, it had been used and applied to Ebola. And so there was already some software that was tied to some analysis protocols. And very early on, people were able to take those protocols, obviously change essentially the kind of lab side of it in terms of what they were analyzing, what they were essentially sequencing up, And then on the software side, really apply that in a way which enabled people to evolve it, to scale it up, and ultimately to really be building it up in a way that was able to go to huge amounts of scale. From those protocols and from the sequencing, the UK in particular was sort of a big leader here. At one time, at sort of the peak of the pandemic, for every positive sample which was being collected, they were sequencing about 20% of those, which ended up being really kind of millions of samples that were being processed. I thought what was really amazing here was just the collaborative efforts of that. So the ability, not just focusing on necessarily the actual software which is doing the analysis, but even on the infrastructure. And when you have a sort of infrastructure as code, it enabled people to have essentially the analysis being done at the point of sequencing, and that allows people to get very fast results, and that kind of turnaround time from sample to be able to get the results and to be able to then track those results across the country and across the world really kind of enabled people to get a kind of full picture on what was occurring as part of that. Really, since then, it's become seen that there's a lot of value in being able to understand what's happening with, obviously with the SARS-CoV-2 variants, but also more broadly. And it's all the application of sequencing can then be applied more broadly to other diseases. So if we think about one of the famous examples, which is used now, is around thinking about something like seasonal flu as like a weather map, where you can imagine the various variants that are around. And because the cost of sequencing has decreased so much, we can really start to be applying this much more routinely in the clinic, and for us to get a better picture of what's sort of taking place out there in the world.
I like this idea of the cost reduction and greater applicability in clinic. As you say, it seems to bode well for the future. But what about the barriers? We discussed earlier, or mentioned earlier, that there are quite a few barriers to entry, aren't there?
If we think about what was, say 5-10 years ago in terms of what was required, very often people were building it themselves. And what I mean by that is that if you were a bioinformatician in a lab, maybe you would have some software you could download, but you were essentially responsible for going quite deep into the details of how that software works, of parameterizing it, of almost like doing the plumbing between the different steps of that, setting up that infrastructure. And it requires really a lot of time, a lot of expertise. And in some ways, you really are just relying on the fact that you're going to get it right yourself within your team. The real benefit of being able to collaborate by having people working on the same code base, by having openness in the actual tools and methods that are being used, is that you get so many sets of eyes on that. You get domain experts coming together to work on a similar code base. And you also get really folks who can build that infrastructure in a way which you can spin it up in the cloud. And in some ways, that reduces both the barrier to entry, because in some ways you're now using and at least kind of using the work of others who are around you. But it improves the quality, because it means that you have domain experts who have really seen this stuff many times before, who can check, and that in some ways lowers the barrier to entry. If we think about something like in terms of genomic surveillance, so I mentioned that we're talking about, let's say, SARS-CoV-2 sequencing. As part of that, many of the folks that are coming into it, but maybe they're coming from a more biology background. Myself personally, sort of came from the lab and sort of did some more, I guess, sort of formalized training in terms of going onto the computational side. But it's definitely a barrier. It's the part where we see most folks coming from. They're often coming from a lab background or a biology background and trying to pick up those skills. So anything you can do to support that journey, and not just lower the barriers to entry, but also to really improve the quality of the analysis which takes place. How we do that? There's many, I guess, approaches to doing so. One of the sort of best ways we've seen this happen is through a community called NF Core, which started around 2017 or so. And what NF Core is, it's a collection of pipelines that have been built by various people. There's around 100 of them, all doing various things, for example, RNA sequencing or variant calling. There's even some pipelines in there for doing analysis of satellite imagery data. But what it means is there's a community of 13,000 people across the different Slack channels who are working together who are building the materials that are required to get people started from very early on, with sort of very little experience, let's say, and both how to use the tools and then sort of understand it and then to be able to contribute back as part of that. And those kind of community ideas, that this concept of sort of open science going beyond just the code, but the open science meaning that you can build really software applications, you can build analysis, you can open data, you can create open infrastructure, lowers the barrier to entry significantly and making it much more easy for folks to join in and then, ideally contribute back. And then we sort of, everyone sort of benefits from that.
That's absolutely fascinating and amazing. And that, again, it's heartening that there is this development that's changed through notions of openness, collaboration, connectivity, community, essentially, to build into the future. And on that notion of the future, what does it hold, Evan, in terms of genomic surveillance beyond 5 years, beyond 10 years?
Well, it's somewhat crude, but my PhD supervisor always said that he'll know when it's really happened when everyone has a sequencer in their toilet. Oh. And the kind of concept here is that sequencing will be really everywhere around, and that this kind of cost of sequencing will reduce significantly. What that really leads to is it's up for interpretation, I guess, in terms of how that plays out. One thing that's not going to change is the requirement for the analysis of the information. It's not just reading the information, but it's also being able to write information. And there's many those sort of startups as well as more established companies now who are developing methods for writing information into DNA. And then we've also seen with the development of more personalized medicines as it relates to the development of personalized mRNAs or specific CAR T therapies, where all of the sequencing information becomes really relevant for being able to really improve in terms of drug development, in terms of new diagnostics, and be able to really make this much more rolled out. It's still very expensive, many of these treatments, many of this surveillance, but we see this kind of constant reduction in price just as the cost of sequencing has reduced so dramatically over the last 10, 15 years. That the same thing will happen as it rolls out in terms of the kind of more broad technological applications of this.
As I say, absolutely fascinating. Thank you, Evan. It's been a pleasure.
Thank you so much.
And so that concludes another episode of the Pharma Forum podcast. You can find more information about this episode, including a download link and information about previous installments at pharmaforum.com/podcasts. The Pharma Forum podcast is also available on iTunes, Spotify, Amazon Music, Podbean, and pretty much wherever you get your other podcasts. Just search for Pharma Forum and press subscribe. Of course, don't forget to visit our website itself, where you can sign up for daily and weekly news and analysis bulletins. And do follow us as well on X or Twitter and LinkedIn at Pharma Forum. That's all for now. Thank you for listening.